Now we're going to shift gears over to really a very specific set of data around the clinical experience with the TRK inhibitors. And Tony, we've tagged you to start with a fairly obscure abstract that just came out around biliary cancers. I know a disease you like at the recent ASCO GI meeting. I think the last face-to-face meeting that will be held on the planet. But maybe talk a little bit about what that showed in biliary tract cancers.
I mean, I think, you know, just to focus on the gist of this, this was primarily focused on IHC, but also had NGS confirmation of the IHC. Just shows that there is a relative concordance between the two, the IHC and the NGS. Although again, as we've heard, you know, we all favor an expanded genomic sequencing because we're looking for a lot more than just the one, at least in specific tumors, such as biliary tract cancer, where we know FGFR is relevant. We just had an FGFR agent approved for FGFR fusions. We have IDH alterations. We have HER2 amplifications. So we want to really have the full gamut, not just NTRK. What this study mostly shows is that these NTRK fusions in biliary cancer are present in about 0.7% of all patients. And that aligns with, the rarity of these fusions, which is pretty consistent for most cancers. I mean, we know there are some cancers that have these fusions at a much higher rate, but in GI malignancies and others as well, the 0.7% seems to be consistent. And that's pretty much the gist of this trial. It confirms the rarity of these fusions in a rare tumor like this.
And biliary tract for us is a target-rich place, but it's about the same as we're seeing in other cancers as well.
Yes. And if you find it, you're going to go after it because again, these tend to be driver mutations, and the responses from the studies look pretty interesting.
Mark, we've tagged you with the job of kind of setting the stage of larotrectinib and its pathway and eventual approval. So maybe just set the stage for us about the trials and inclusion criteria and how that went.
Yeah, the original trial that led to the FDA approval was a trial that selected prospectively patients that were TRK fusion positive. It was actually a combination of three different studies: a phase 1 study in the pediatric population, as well as a phase 1/2 in the adult population, and then a phase 2 study that involved both adolescents and adults, again with NTRK fusion positive cancers. There were 17 different cancers that were included in this. There were only 4 lung cancer patients. I think talking about the bile tract, I think there were a couple of biliary patients in this. Patients ranged in age, this was fascinating, from 4 months to 76 years. The interesting thing is that the response rate objective by independent radiologic review was 75%. So the vast majority of patients responded. They had a couple of patients that were early progressors, and it's not clear that they were true positives. There were some issues about the testing that was done. They also had a patient that progressed early that had received a prior NTRK drug and had a resistance mutation. So that patient would not have been expected to respond in this particular setting. It was remarkable in the sense that no patient had the drug discontinued because of toxicity. So very well tolerated, very few grade 3 or 4 adverse events, mostly were grade 1 or 2. So kind of the best of both situations. I think in the most recent update, others can correct me if I'm wrong, but I'm not quite sure the median duration of response has been reached at this point. So it's one of, I think, the second drug after the pembrolizumab MSI high indication that was tumor agnostic, but linked to a molecular alteration regardless of your histology. And really, I think, an important step in how we think about cancer and get drugs approved.
As another member of the panel who graduated from fellowship in the year beginning in 19, I think I remember being scolded for suggesting that we wouldn't need phase 3 randomized trials if the benefit was obvious.
Yeah.
And that it would transform drug development. And what I'm so pleased of is, even though I got beat up pretty bad for, no, you're wrong back then, this is the way it's going. And maybe comment a little bit about how this has changed, like, our whole perspectives of cancer and drug design.
Yeah, no, it's been fascinating. I remember you, actually, John, you may be the only one on the call that remembers a gentleman named Joel Tepper. Joel Tepper, a radiation oncologist at University of North Carolina, would always say, I'd like to do a phase 3 trial that required only 50 patients, because the effect was so big that you didn't need 1,000 patients to, to prove the benefit. But again, the larotrectinib indication is a growing list of drugs that should be approved for these molecular subsets of patients that can be clearly defined. In lung cancer, we have the EGFR and ALK experience, where we actually went through doing a number of randomized trials to show that when you have a target that's targetable with a targeted therapy, either an EGFR or an ALK drug, it's better than chemotherapy. Do we have to do that for NTRK? Do we have to do that for ROS1? Do we have to do it for every single molecular category that we have to prove that it's better than platinum? Why are we in love with platinum-based therapy in this setting? If you have a target where it's driving the cancer, and with the targeted therapy, all the response rates are north of 50%, and the durability is 3 to 4 times longer than we see with chemo.
And the feasibility, this wouldn't be feasible with something so rare to randomize it against something. So to me, the wisdom and the flexibility of the FDA has been terrific in this space.